Thank you very much, Nicola. I come under the category of scientifically illiterate, so I'm all ears. So welcome to the annual Gretchen Amphlett Lecture. This evening, along with you, I'm looking forward to hearing Dr. Stott and Professor Bandmann on their... My involvement with the Parkinson's research began with joining the Research Support Network, and as is the nature of these things, I was then tempted into the East of England Research Interest Group, whose aim is to raise awareness of Parkinson's research and help researchers with their work. I won't say any more, but please talk to the members of this group this evening who are around, so if you need to know anything about what we do, they are the experts. Even with his involvement in research, I was surprised and honoured to have been asked to introduce the proceedings. And as a non-scientist, I admire those who understand the weird workings of our bodies and try so hard to fix them when they're not behaving as they should. I am also a bit like a faithful Labrador. I often do not understand much of what they say, except when I'm told to walk and sit. But I'm nevertheless full of a willingness to please. I'm delighted to report, however, that Parkinson's UK leads the way in demystifying science and engaging us, for whom they are working so hard in its development and in its challenges and triumphs. We do not have non-success. We merely have pointers to a different route to a cure. The burgeoning number of committees and steering groups where researchers and Parkinson's people meet and listen to each other are, I am told, exemplary. So can they please give themselves a clap? And a pat on the back. And while I'm thanking them, can I add in that what they do for us, nationally, regionally, and locally, is a lifeline to the Parkinson's community. I don't particularly want to have Parkinson's, and this will probably be no surprise to you, but if I'm stuck with it, I couldn't ask for a nicer bunch of people to support me. But it is the determination to communicate the progress that we celebrate here this evening. This is a bit like Pilgrim's Progress, but we don't go anywhere near the Slough of Despond, and we keep our sights on the wicket gate which leads to ultimate success. Since writing this, I looked up the definition of Slough, and it means a situation characterised by lack of progress or activity. But I think we can honestly say we don't have that. Perhaps I'll get a little personal here. As a non-scientist, how do I react to the way my own body, without asking my permission, which is extremely rude of it, declines to obey my instructions and will not always do as it's told? As a relative newbie to the Parkinson's world, diagnosed a mere 4 years, what is my stance to the condition's obvious desire to stop me enjoying myself? Well, I really don't like it. So I've adopted the mantra, while I can, I will. This means, in essence, that when someone says to me, would you like to? I've usually said yes before they finish the sentence, which is probably one of the reasons why I'm standing up here today before this distinguished audience. The upside of this is that I often find myself doing things I really enjoy, but didn't think I could ever manage. And yet I do, and so my non-scientific and completely anecdotal advice to fellow Parkinson's people is to have as much fun as you can. It's not always easy, but it's really worth the effort, and I think you really, really try. It's good for you. So this introduction is not all about me, but more about us. I will turn to Dr. Stott's and Professor Bandmann's illustrious predecessor, the good Dr. Parkinson himself. His practice might have been lacking in expertise of today's genetics and sophisticated chemical compounds, but he had something equally, well, actually more important, in some ways, I think, in abundance: a love of humanity and a will to find a way through. To quote from his seminal essay on the shaking palsy, there appears to be sufficient reason for hoping that some remedial process may ere long be discovered, by which at least the progress of this disease may be stopped. I believe that's what we're here for today. And further, he declares, with reference to researchers, by their benevolent labours, the condition's real nature may be ascertained, and appropriate modes of relief, or even of cure, pointed out. To such researchers, the healing art is already much indebted for the enlargement of its powers of lessening the evils of suffering humanity. Little is the public aware of the obligations it owes to those who, led by professional ardour and the dictates of duty, had devoted themselves to these pursuits. I think if you translate this into modern English, it's good on you, researchers. You're doing a grand job. So let me applaud the researchers and the clinicians for what they've achieved so far, and thank them in advance for their efforts towards the cure they are going to find for me and my friends. The only thing I would ask is, could you do it sooner rather than later? I really don't want to wait another 200 years. And so let us sit back, as they say, and enjoy the speaker's thoughts and their efforts on my behalf. Thank you so much.
Firstly to Parkinson's UK for inviting us to come and speak, and also for partly funding the research that we're going to be showing you tonight. We're here to have a discussion about how simple tests are going to potentially revolutionize the way we diagnose, monitor, and treat Parkinson's. And what I would like to do tonight is to show you some of the initial results that we're getting from the Parkinson's UK BioRx 2 breath analysis study. But one thing I would not want you to do tonight is to go home thinking that this was all my doing. I'm merely a minor player in this story. So with that in mind, I'd like to start off by introducing you to some of the... This is not working at all. I'll move over here then. Those introductions start with this lady here. Now, this is Dr. Orna Barash, and she is one of the Israeli engineers that I work with on the Parkinson's UK BioRx 2 breath analysis study. And it was the first time that I met Orna that I truly came to appreciate how powerful the technology we're working with could potentially be. You see, she had come to Cambridge to set up the device, and this is the device here. I've put a pencil in the picture just to show you the scale of it. This is the breath analysis machine. And she was... right, as easy as that. So she had come to Cambridge to me how to use it, and I wasn't really listening. I was just staring at it and gushing on about how amazing it is that we can even do this. And it is an amazing piece of kit. You breathe into this mouthpiece here like you're blowing on a whistle, and inside the machine, the chemicals in your breath are separated out and analyzed individually, and then all that information is sent via this cable to a nearby laptop, and it can be further analyzed and compared with other samples taken from other individuals. And I was gushing on about how amazing this is, and Orna was amused by my enthusiasm, but she was very quick to point out that this is only the first step in the development of this technology. This is just the first stage in the evolution of it. I'm a pretty simple country boy from the backwaters of third world New Zealand, and I thought this was a pretty good end product in itself. And I said, whatever do you mean? And that was when she reached into her pocket and pulled out her cell phone. And she explained to me that at this moment, they're trying to shrink all that technology down to something that will fit into the bottom of this device here. And so in the future, every time you make a phone call, the machine will be analyzing your breath. And to do this over time. Each time you make a phone call, each time you call a friend or family or call for dial-up pizza, it'll be analyzing your health, and it'll be able to send that information to the hospital or to your GP or your doctor, and they'll be able to have an electronic file on you. Enough red flags pop up on that file, they'll send an ambulance out to get you. Even if you don't realize that something. So this is where you have a small test, a simple test, but there's this enormous potential for it. Now the obvious question is, how does this relate to Parkinson's? And that's where the second character in my story comes into the picture. This dodgy-looking fellow. This is a large research group here in Cambridge University, where we focus on both clinical and lab-based research on neurodegenerative conditions, particularly Huntington's and Parkinson's. And tonight I'm just going to be talking about Parkinson's. Now, everyone in this room is relatively familiar with the condition. For the one or two of you that are unfamiliar, Parkinson's is the second most common neurodegenerative condition after Alzheimer's. It is characterized by three, excuse me, resting tremor, slowness of movement, and rigidity, as well as a variable host of non-motor features, such as sleep disturbance, loss of smell, gastrointestinal problems, and in some cases, cognitive issues. About 1% of the population over the age of 60 are affected by it, and at present, there are no curative treatments, and it's pretty fair to say that our methods of diagnosis, and in particular our methods of assessing people over time, are very basic at best. We desperately, desperately, desperately need better methods of assessing people over time, particularly from the standpoint of clinical trials. And that's where the third character in my story comes into the picture. This is Professor Hossam Hayek. And a lot of the credit tonight belongs with him, because he is the inventor of the breath a large research group here in lovely Haifa, Israel, at the Technion, which is the Israeli Institute of Technology. And a few years back, he and his team started collecting breath samples from thousands of people with all sorts of different medical conditions, dozens of medical conditions. And they started questioning whether they could differentiate between those medical conditions based on the chemicals that are in our breath. And they presented a lovely example of this where they took 30 people with Parkinson's and 12 people without. And just by analyzing the chemicals in their breath, they were able to differentiate between these two groups to an accuracy of 80%. That is to say, 80% of the time when they took a breath sample from these people, from one person in the study, they could say this person has or hasn't got Parkinson's. And that's pretty impressive. But then the next year they went one step further. And they took a group. So this is a spontaneous version of Parkinson's that can't be attributed to any genetic or environmental factor. And they compared them, those 44 people, with 16 people with non-idiopathic Parkinson's. So these have a genetic or an environmental factor, some sort of causal element. And they could differentiate between these two groups to an accuracy of almost 90%. Now just stop for a minute and appreciate that both groups have got Parkinson's. And all they were doing was looking at the chemicals in the breath. We thought this was a pretty good trick. So we said, let's collaborate. And that gave birth to the Parkinson's UK BioRax2 breath. Rather than just simply doing this on a large scale, which we're doing with 200 people with Parkinson's and 100 without, we said, let's focus in on the Parkinsonian population and see what happens when we follow these people over time. So it's a longitudinal study, multiple samples. I've got one gentleman who's given me 5 samples now. He claims he's sending himself to Israel one breath at a time, hoping to retire there one day. And what we'll do is we'll compare all the clinical data that we have on these individuals with all the breath samples that we have and see if we can't differentiate between different types of Parkinson's. Tease out maybe people that are tremor dominant versus people that have got more rigidity or gait issues. Because our situation at the moment is that we have this kind of singular group that we call Parkinson's. And what we really need to do is start to differentiate between the different types and focus treatments and therapies on those groups, those subpopulations of people. Now, some of the more curious members of the audience might be wondering what on earth are these chemicals that he's talking about in my breath. When you breathe out, about 80% of what you breathe out is nitrogen, 16 to 15% is oxygen, dioxide, And then this small yellow slice has the very technical term other stuff. Your body has only basically 4 ways of excreting waste. You can urinate it, you can defecate it, you can excrete it, or you can breathe it out. And a certain portion of what you breathe out are called volatile, are ubiquitous, and they're extremely numerous. And they're basically chemicals that are vaporous at room temperature. For example, some of you might recognize chemicals like ethanol and methane, for example, methanol, excuse me. And what Professor Hayek and his team have been able to do is, if you consider each of the volatile organic compound, they've been able to find compounds that are specific to all of the conditions that they're looking at. But then there are compounds that are specific to only certain conditions. For example, here, lung cancer has an average level of this particular volatile organic compound, whereas ovarian cancer doesn't. And you build up a tapestry that is unique to each of these conditions. Here is idiopathic Parkinson's, and just below it is atypical, non-idiopathic Parkinson's. So we've now got to the point with the study where we have a lot of data. We've got a lot of data, we've got a lot of breath samples, and we're doing the analysis. And the analysis is being done in 2 different ways. Firstly, a biased way, and then we're going to do it in a characteristic that we're aware of, something that we can know and measure very easily, and try and see if we can find associations with a particular combination of chemicals in the breath using that approach. So we've taken 3 analysis. Firstly, the duration that a person has had the condition. So how many years has it been since you were diagnosed? It's very easy to measure. Verbal fluency is a measure of cognitive ability. How many words can you think of in 90 seconds, starting with the letter X, for example? What? And finally, an assessment of mood. So we've used a survey that people fill out themselves. It's a multi-choice questionnaire, and indication of how this person is feeling. Are they happy? Are they sad? And what we find, measure. We can clearly differentiate between folks who have been diagnosed for less than 6 years, for example, and those who have been diagnosed for more than 6 years. And what's interesting here is that, I don't have the picture to show you, but regardless of how long an individual has actually been diagnosed, we can also separate out how features that is. For example, you might have just been diagnosed, but you might have a very progressive form of Parkinson's, or you might have had Parkinson's for 20 years, and it's a very mild form. We can also put it in the breath analysis. Another of the biased analysis is the verbal fluency. So we have a group of people over here who can give us less than 50 words starting with the letter X, and another group over here who can give us more than 50 words starting with the letter X in seconds. And again, it's a pretty clear differentiation between the 2 groups. And finally, the mood assessment's a little bit muddier. It's not quite as robust as the other 2. This one could be affected on a day-to-day assessment level. So that's the biased approach. And now we're at the stage where we're the unbiased approach. And this is where we feed all the data that we have, the clinical data as well as all the chemicals in your breath. We put all that into an artificial intelligence machine, and we let the machine find what associations might exist. And this might be stuff that's not exactly clear to us, might not be obvious to us, but it'll be interesting to see what results we get. So hopefully, from my brief talk tonight, there is the potential of using this technology, firstly as a diagnostic aid, but also for monitoring the condition over time. And there's also the potential for future research, which could explore early diagnosis potential of this technology. How far before a person is diagnosed could we pick up some of these chemicals in the breath? And also, where in the body are chemicals actually coming from? This could tell us a lot about the condition itself, the biology of it. And obviously, such knowledge would and new methods of monitoring Parkinson's over time. And for the folks in the audience who were skeptical when I said about the cell phone idea, this is Professor Hayek. This photo was taken a couple of years ago, and this is the original prototype component. And this is now, the latest rendition of this technology is now in clinical trials. So don't be surprised in the near future if you're on the phone one day, your new iPhone, and there's a knock on the door, and it's in the hospital. And with that, I'd just like to thank Professor Hayek and his team, and Professor Barker, Birax, and Parkinson for funding this study. Thank you very much. Thank you. Thank you. And what I'd like to do now is hand over to my glamorous second speaker, Professor Bandmann from the University of Sheffield. Thank you.
Good evening, everybody, and I would like to express my sincere thanks as well for inviting me tonight. I'm really blown away, I have to say, by the activities of this branch, by the activities of all the people who've set this up some years ago, and I'm honored, and I feel this is a very important part of what people need to do who carry terribly pompous titles, such as being a professor of this or professor of that. We need to reach out, and we need to try and explain what we do. I love doing what I do, and I hope that this will also come. So what my talk is going to be about really is, in a nutshell, how can we do better in the future? Because let's just remind ourselves again what the focus of this session is, of this lecture is tonight. How can we do better at choosing the right patients for the right therapy? So we all know that there is a dire clinical need for us to do better. Parkinson's disease continues to be relentlessly progressive. We still don't have any disease modification. We still don't have any medication which would modify the progress of the disease, which would slow down disease progression. And even if you're a politician who's not in the slightest interested in reducing the suffering, maybe there are politicians out there who are like that, even if you're one of those, you should still spend a little bit of money now on research in Parkinson's disease, or ideally even a lot of money, because if we don't crack the problem soon of neurodegenerative disorders, then this will break our societies from the financial point of view. Because there are more and more old people in the society. This means more and more people will develop illnesses such as Alzheimer's or Parkinson's disease. And this does have, apart from anything else, also massive cost implications. In the US alone, Parkinson's disease costs the US society about $10 billion a year. So we badly need to crack this problem. So what do I mean when I use the term neuroprotective treatment or neuroprotection? So it really does matter that we explain these words very well and that we all agree on how we should be using them. So my obviously refers to nerve cells, i.e. those which normally die in Parkinson's disease. And protection is what it says on the tin. It's not the same as a cure. It's not the same as a miracle or a resurrection of the cells which have already died. So my definition of what this is all about is I want to find a drug or drugs which will slow down the cell death, which will make sure that the number of cells which die every year will decrease. and also, and that's a really important point, that the remaining cells are as well functioning as they can possibly be. So the earlier we can start patients on medication, which will have this effect, and the more powerful these drugs are, the less a patient will be affected in his health, well-being, and happiness by Parkinson's disease. But so how come that all previous attempts have failed? How come we still haven't got neuroprotective treatment for Parkinson's disease? It's certainly not because people haven't tried. For me, a key reason why we've failed so far is because Parkinson's disease is not a disease entity. So if you just imagine that all these people are patients who've been included in a drug trial where yet another drug was tested for its effect to slow down Parkinson's disease, then some of them will have only developed the disease because there's something wrong with one of their genes. But that's quite rare, at least in the UK. Only 5, maybe 10% of all patients will have developed familial Parkinson's disease. Others may have only a high dose of pesticides or herbicides. But again, that's quite rare. The current thinking is that the vast majority of people who later on go on to develop Parkinson's disease will have been born by genetic susceptibility or vulnerable factors, and they will then subsequently be exposed to exogenous factors, such as exogenous toxins. And it is the combination, really, of these inherited risk factors which make you more vulnerable, and subsequent exogenous factors, probably environmental toxins, which really then give you the disease. But there's so many other things we actually don't know yet. So, for example, how important is it whether you've eaten your greens or not? And if it is important when you're in your 20s? Or is it only really important when people are, approaching the age when one typically develops Parkinson's disease? What about all the other drugs people are using? So, for example, there is now reasonably good evidence that those people who have taken quite a lot of ibuprofen, an over-the-counter drug for musculoskeletal pain, I'm sure many of you have been on it yourself, are probably at a reduced risk of developing Parkinson's, which in its own right makes it also somewhat plausible to assume that these drugs may slow down the progression. But of course, if you want to reduce the risk of developing Parkinson's as much as possible, put yourself on a Tom Waits diet. Make sure that you smoke like a trooper, and make sure that you drink coffee as much as possible. Why is that? Why are these two factors the only factors which are established beyond belief which reduce your risk of developing Parkinson's disease? We have no idea. So the buzzword in 2018 for Parkinson's disease, but also really for many other illnesses, is personalized or stratified medicine. What do we mean by this? It's now quite clear that disease in different people. So, and the key 4 mechanisms the field largely agrees on are the following. It's inflammation, it's mitochondrial dysfunction. The mitochondria are the cell batteries which generate the energy in the cells. It's lysosomal dysfunction, so the lysosomes break down the cell garbage. If a cell can no longer get rid of the cell garbage, then the cell will essentially pack in and stop working, and linked to that, it's protein aggregation. So our cells synthesize more, make more and more proteins, but if they make too many of these proteins, if they can no longer be broken down properly, then that's also bad news for the survival of the very cells which die in Parkinson's disease. There's now very strong evidence when you look at the genetic level. There's very strong evidence for an involvement of all these mechanisms when you look at the brain of a patient with Parkinson's disease after they've died under the microscope. But, of course, we now need to take the next step. Of course, we now need to get better at working out whilst the patient is still alive, whether the Parkinson's disease in Mr. Miller is predominantly due to a problem. Disease in Peggy Smith is predominantly due to mitochondrial dysfunction, or whether it's a combination of all these things. That's what it's all about. So the next big buzzword is biomarkers. We've just heard a wonderful talk about this, a very promising new approach. And of course, many other people are working on complex biomarker research approaches. But again, it's important to emphasize, we do not have a biomarker for Parkinson's disease yet. So what have we done? How did we try to tackle this? So we set a disease center, and we looked at skin biopsies. So we took skin biopsies from 100 patients with sporadic Parkinson's disease, 50 controls. They were recruited from these two sites. Sheffield recruited about a third of the research participants, Oxford about 2/3. Age was about 60, and all patients were screened for the two common mutations, LRRK2 and GBA1. And the next thing we then did was we analyzed. It's really straightforward. I've had one myself. I can show you the scar later on, but I'm afraid it's not very impressive. And then we looked at the different cellular readouts. We looked at the function of the mitochondria, we looked at their shape, and we also looked at the lysosomal function in all these cells. And remember, these are humble skin cells, so we didn't do anything clever with that. We didn't transform them at that stage into what's called inducible stem cell-derived neurons. These really are just humble skin cells. And in a subset of patients, we also investigated whether Parkinson's disease patients may be more vulnerable to toxins. And what we got for all these different readouts was exactly the same pattern. I'm just going to show you the lysosomal data. So I'm going to take you through this slowly now. So up here, you can see an image of one of the cells we looked at. So the big blue blob is the cell nucleus, and all the red dots are the different lysosomes. So remember, this is a cell mechanism which helps to break down the cell garbage. And to analyze the data, you then binarize this image into black and white, and that makes it easier for special software to then quantify the dots. And that's what you see here. So on the left, you see the control group, and each control person is a blue dot. And on the right, you see the patients, and each patient, again, is a red dot. So this work took several years, funded by Parkinson's UK. And you can see several things here. So the first thing you see is there is very clearly a very marked overlap between patients and controls. So it's by no means the case that every single patient with Parkinson's disease has a massively increased number of lysosomes. If you look at the data a bit more closely, then you will see that this dotted line represents 2 standard deviations, and this bold dotted line represents 3 standard deviations. What you can see here is that there are about half a dozen patients who have a markedly increased number of lysosomes, increased by more than 3 standard deviations. And we then selected these patients, their cell lines, and looked at them in greater detail. We then looked at the function of the lysosomes. And as you can see here, there is, despite this only that now being a comparatively small group of half a dozen patients, a definite decrease in the function of the lysosomes. So we think that this indicates that these patients may be good candidates for the next round of drug trials. So if the next drug wants to be tested by us or others, which has a particularly marked rescue effect on the lysosomes, then we postulate that these patients are much more likely to benefit from this lysosomal rescue drug than, say, these patients. Stratification work linked to the drug discovery work we've done at our wonderful Sheffield Institute for Translation Neuroscience. So some years ago, we started working on the mitochondria, the power generators in the cell. And at that stage, we used skin fibroblasts from genetically stratified patients with Parkinson's disease only. So all these patients have mutations in a gene called Parkin. And you don't need to know what mitochondria does give you a very good idea whether there's something wrong with the mitochondrial function in these patients or not. And as you can see, there's something very wrong in this patient group. So this was interesting in its own right, but it also gave us a really big therapeutic window to screen drugs against. And that's what we then did, again with a grant from Parkinson's UK. We then screened 2,000 drugs in patient tissue. So this was the first ever drug screen in Parkinson's disease patient tissue, and I believe it's still the only screen which has ever been carried out in patient tissue, at least the only published screen. So this was done by Heather Mortiboyce, wonderful scientist who started off as a postdoc in my group and is now a Parkinson's UK senior fellow. And we took these 2,000 drugs through a multi-step process and ended up with a bunch of very promising drugs. And the most promising one, we think, is a drug called ursodeoxycholic acid, or UDCA. So this is a really interesting drug, because we know that from work others have carried out, that it does have a very strong protective effect in all traditional toxin-induced model systems for Parkinson's disease. We know that it's safe and tolerable in a different neurodegenerative disorder, motor neuron disease. It has already been in clinical use for a completely different disease, for a particular type of liver disease, for several decades. That's why we know that it's safe, which also means it's an ideal candidate for the so-called drug repositioning strategy, which means you grab a drug which is already on the market, which has been used for decades for a completely different illness, and then you try and find out whether you actually can slow down disease progression in Parkinson's disease by putting patients on this drug. And I'm excited, very excited, that now, 5 years later, and on my 7th attempt, I finally managed to get some money from the J.P. Moulton Foundation to take this drug into an early clinical trial, a proof of concept study. This is all very well, you might say, but hang on a second, didn't you say early on that you did your clever drug scheme and patients were stratified, which were stratified, and wasn't that only patients with that Parkin mutation? Yes, absolutely right. So how can we be certain that UDCA really works in the much more common sporadic form of Parkinson's disease? Of course, we will only know this for certain once we've done the clinical trials, but of course, we've now also tested UDCA, again in the skin fibroblasts from patients with sporadic Parkinson's disease. And as you can see here, again, there's a marked decrease. It's by no means as pronounced as in the genetically stratified group, but there clearly is a decrease in some patients with sporadic Parkinson's disease. If you put the patients back on, if you put the cells on UDCA treatment, then you normalize it. And we think this is extremely encouraging. So what I've just talked to you about is the past, really. So this was the first drug screen we did, and we're now taking this drug through to a clinical trial, and that's, I think, exactly what academic scientists, academic clinicians should be doing. But this has now led to a wonderful development in that we're now carrying out another 3 Parkinson's disease-focused drug screens at Sheffield or from Citran. 2 of these programs are funded by Parkinson's UK. The third one is commercially funded, which are also very exciting, and the methodology kind of overlaps with the methodology I just described. But we also need to look into the future, and I think the future will really be for us to do less and less at the bench and more on a computer. So the future has started already, because the future really is in silico drug screening. So what do I mean by this? It really means that we do as much drug discovery on the computer and as little as possible in the lab. And there are different strategies how we can do this. So we set up a collaboration with our extremely powerful bioinformatics group at Citron. And this is just to remind you quickly of basic biology. We look at the transcriptomic profile, again, in these skin fibroblasts. And just a brief reminder, so DNA is, of course, the inherited genetic material. It's, of course, transcribed into messenger RNA, which is the template for the real thing, so to speak, for the proteins, the enzymes, and so on, which then do all the work in our cells. And all our 20,000 or so genes in the body are up or down regulated to different extents. If you have an illness such as Parkinson's disease, you can then compare the transcriptomic profile in patients with Parkinson's disease to healthy controls, and you get a so-called heat map. So the upregulated genes are then green, and the downregulated genes are red. And you can then feed these data into clever software programs, and you get a disease signature. So certain genes, and really doesn't matter at all which genes we're now talking about, certain genes will be upregulated, others will be downregulated. Okay, that's just the first step. The next step is, fortunately, other terribly clever people have assessed thousands of drugs for their effect on the transcriptomic profile. So you can then feed the patient data into the databases, the computer databases, to identify drugs which have had exactly the opposite effect to the changes you saw in the patients. And the net result should be a normalization of the gene expression profile. And that, at least that's the idea, should then tell us that the drugs which do this, which normalize the gene expression signature, may be good drugs to take into a clinical trial. So we've just completed the first analysis. So this was a study funded by the Michael J. Fox Foundation, and the results are astonishing. And I'm really not exaggerating here. It's really exciting, but this is, again, just in genetically stratified patient fibroblasts. We're now doing the same thing again with sporadic patients. Of course, remember that we have 20,000 genes or so. Of course, we can't find drugs which correct every single gene which is dysregulated in Parkinson's disease. So it'll be particularly powerful if we attack the nodes, because all these genes, they work together. They form networks. Sometimes it's just a few. Sometimes it's thousands of genes which work together. So, of course, it will be particularly efficient if we attack the nodes of these networks. And that also holds great promise for future powerful Parkinson's disease drug discovery. Well, this is all very nice, you may say. But, for God's sake, what I really want is, a better idea whether the next drug, Roger Barker or whoever, whom I think very highly of, by the way, he's a great guy, will really work or not. Because let's face it, all of you have Parkinson's disease. All of you who know people who have Parkinson's will know other patients who have responded in a totally different way to exactly the same medication. And sometimes there are obvious reasons. Sometimes it's simply how long a patient's had Parkinson's disease for, or whether they have other illnesses, etc. But in most cases, quite frankly, we haven't got a clue, right? So that's, where we are at the moment. And let's make today is very much still 19th century medicine, right? Things have not moved forward greatly since James Parkinson, maybe a little. But, it is still try and error. That's really what it boils down to. And we must never, ever be complacent about this. It's just not good enough. So how could personalized medicine help us to get a better idea whether a patient will really be responding to drug X or not? So again, there's some promising first signals coming through from the literature. This is a busy slide, and it's a bit complicated. I'll just take you through it slowly, because I think it's an important message. So down here, the x-axis just gives you the number of weeks patients have been on a drug called rasagiline. Many of you will have heard of it. Some of you may be on it. It's one of the enzyme inhibitors, which essentially slows down the naturally occurring breakdown of dopamine. And here is the severity, Parkinson's disease severity score. So, and of course, if you start a patient on symptomatic treatment, then you expect the severity score to go down, so to speak. If there is no effect, then you expect the patient to essentially remain the same or get worse over the next year. So, and you can actually now assess the particular gene variants within the very enzyme which rasagiline inhibits. So let's just say there is a good and a bad genotype here, right? And you can, at baseline, assess whether the patient has genotype A or genotype B. And then, according to this study, you can actually predict whether the patient will respond to rasagiline and get good motor response, or whether nothing happens at all. Now, you may say, oh, the data are not that impressive, are they? I agree. They're not desperately impressive. But, this is just one study, and it's just the beginning of, what we will all hopefully be doing much more of in the future. So what we do at the moment is just not good enough. At the moment, as you know, when you're newly diagnosed with Parkinson's disease, you get half an hour with a consultant, maybe 45 minutes if you're lucky. And then you started on L-DOPA or whatever or not, and you come back in a year. In the future, I think we'll have a completely different scenario. Once a patient's been diagnosed with Parkinson's disease, you will be admitted to the wards for a week, right? And then we will throw the book at you, right? Your entire genome will be sequenced. You will have a whole body scan. Your breath will be analyzed. You will have a skin biopsy taken. It will be transformed into dopaminergic nerve cells. We will tell you by the end of that week, brackets, it may take a little longer, brackets, which type of Parkinson's you have. We will tell you by the end of that process which drugs we have already tested in your own tissue, and then we'll start you on that treatment. That is the future, and it will happen. And so many of my Parkinson patients continue to do amazing things, and I want to help them. I want to do my bit to make sure that they can do these things as long as possible. So this is one of my patients who cycled from London to Paris last year. Look at the wonderful dexterity. I'm a bit of a train nerd myself, so I thought I had to chuck that in. Look at the wonderful dexterity, which is reflected in this wonderful train set, from another patient with Parkinson's disease who's under my wings. Look at this wonderful gentleman, who loves spending time with remote-controlled aeroplanes. Not such a good idea to have, not such a good thing to have bad tremor when you control one of these things, he told me. So, I look at this gentleman with his lovely TR6. You want him to be safe on the road, and he wants to be safe on the road too. We need to make sure that all these people, that everybody, will do as well as possible for as long as possible. That's what neuroprotection is all about. Better understanding of the cause of Parkinson's disease in every single patient will help us to find treatment which will stop the disease from getting worse. I would like to acknowledge the people who've done the work. Also Marcus Keating, who's helped me with some other research, our collaborators from Oxford, and also the funders. I've mentioned Parkinson's UK, the Michael J. Fox Foundation already. I'd like to express my gratitude as well to the JP Moulton Foundation, and also to our now NIHR-funded Sheffield BSc for Translational Neuroscience. Many thanks.
UDCA and how it's being used in liver patients. So do we know anything about the extent to which that group of people have gone on to develop Parkinson's?
So, is this? Oh, okay. Can I use this? Okay. So we tried to look into this, but unfortunately, this is quite difficult due to several confounding factors. So to begin with, it's a particular liver disease called primary biliary cirrhosis, or PBC, or cholangitis now, which is really quite rare. Interestingly enough, it's caused by an autoimmune process which attacks the mitochondria. So there's a clear link to Parkinson's disease as far as that's concerned. But, you know, the vast majority of patients who develop the disease, 9 out of 10, are women. In contrast, Parkinson's is, of course, slightly more common in men. So just because of these few factors, we simply don't have a large enough cohort of patients on UDCA we could analyze for the risk of developing Parkinson's disease later. Point one. Point two, we think that the currently used dose for the liver disease is probably not high enough for the treatment of Parkinson's disease. So patients with the liver disease are only on 10 to 15 milligram of UDCA per kilogram body weight, whereas for Parkinson's disease, you probably need to be on 30 milligram or more.
Okay, brilliant, thank you. So one question here, the idea of separating people with Parkinson's into smaller groups, developing more specific treatments that addresses their particular form, sounds great in theory, but how practical is it? Won't it fragment the market and means that it makes it harder to incentivize people, the market, I guess, to develop treatments for those smaller groups? I don't know, Arthur, do you want to
Sure, yeah.
on that?
Right. Well, that is true. That is a factor, but this is a direction that, in fact, a lot of pharmaceutical development is going because it increases the chance of success. So to have a one-size-fits-all approach with one medication and test it in a large group of people where a significant number might not have the right type of the condition to respond to the medication means the whole trial might fail, and that can be a very large, hundreds of millions of pounds perhaps spent on that program. So many areas, many companies are going in the direction of saying, let's be sure we have the right patient population and the right medication and put those two together. So that might mean, yes, there'll be a number of choices for people. That'll be appropriate for them. Ultimately more success, which is of course better for patients, and that success will also ultimately be better for the companies as well.
Yeah, Oliver?
So, as Claire and others know, my first version of this talk was much longer because I included quite a few slides at the beginning which would have told you about very recent success in a completely different area of medical research, and that's the treatment of non-small cell lung cancer. Why did I include these slides initially? Because between 1970 or so and 2010, there was absolutely no progress whatsoever. The mortality continued to be extremely high for patients with this nasty form of lung cancer. Since then, there has been a fantastic change. And why is that? Because suddenly, it's no longer the case that all patients with non-small cell lung cancer are thrown into one pot. These days, it's broken down to the very last detail. And many patients are now put on stratified medicine, which is exactly tailored for their very own mutation. And that really results in a fantastically improved survival in these patients. And the drug companies love it because they've got a well-identified target.
Okay, thank you. Ruth, how do you respond to Oliver's vision of what it would be like to be diagnosed in the future, spending a week on the ward, having that kind of battery of tests, and I suppose maybe the kind of scary bit, perhaps having a bit more insight into your future, into your particular type of Parkinson's and how that might develop?
I think if you have something that works, you're willing to give up a week of your life to have a much better future life. I think it would be fantastic, because when you look at people who've got Parkinson's and you look around the room at the support group that I'm in, so many people have got so many different symptoms, attitudes towards the condition. If you can actually identify what's wrong with each and every one of them and then treat it, that must be a good thing. That has to be a good thing. And I'd give up a week of my life for that, definitely.
Brilliant, thank you. Simon, how will breath testing cope with older people who have comorbidities? So people with Parkinson's, but also have carcinoma of the bowel or COPD, for example. How do you think that will all play out?
That's a really good question.
It wasn't mine, but someone else.
And I'm not sure how to tackle it. I guess it would be, as with everything else we're talking about tonight, it would be a gradual shift towards individualization. And you'd be looking for these red flags, as I was talking about them, popping up for various conditions within the individual. So if you have Parkinson's and you are developing diabetes, some of these red flags for diabetes would start to pop up. Whether the Parkinson's or another medical condition is influencing or affecting that, at the moment we just don't know. But there's the possibility of it, I suppose.
Okay, and a lot of us on a similar theme have read, have seen the TV programs about the lady, Joy Mild, who can smell Parkinson's, and the work we're now doing with sniffer dogs as well. What do you see the links between the smell of Parkinson's and the studies that you're doing on breath?
So this is the question I get asked everywhere I go and talk about the research. And we're communicating with the groups that are doing this. But the smell that she was picking up is coming from excretion from the skin, primarily from the back of the shoulder blades, I believe. And this is coming from a mucus called sebum that we all express. Whether it's the same chemicals in the sebum as in the breath, we're not sure yet, but I'm looking forward to comparing notes and sharing the data once we have our final data set.
Yeah, okay, fantastic. And maybe a question for Ruth about some of the ethics, I guess, around this. So our mobile phones, perhaps, can identify our health. I'm just thinking, actually, as the chief at Parkinson's UK, we issue our staff with mobile phones. Does that mean I'm the first to know about the health of our staff? I'm just wondering how you feel as a representative of the Parkinson's community about how some of the issues that get unlocked by the kind of explosion of technology in this space.
I think there's always a balance between getting better and who knows about it. And it's a very fine balance. I think if you are working, and I know that if you're working, you either tell, once you say to someone in the company, you've got something, then it's immediately something they have to take notice of. So maybe you don't want to tell people, but then you don't get cured. So, you don't get fixed. So I think each individual has to kind of decide how much privacy they want, whether they want to be not known to have something, not to be known to have something, or they want to be open and above board about it, and then have the facility to get better. I think getting better has to be a good thing, in all cases. And it's not a nice disease, it's not a nice condition, and you don't want to have it. So if you can find a way of being better, as I said before, if you can find a way of being cured or made feel better, then it has to be worth it, I think. And I'm sure I won't be made to vote for somebody I don't want to vote for.
Brilliant, okay. Everybody really, but how many years do you think we are talking about before some of this personalised medicine actually starts to become a reality? Who wants to have a go at answering that? Simon?
So I get asked this question at every place I go to.
I think what Simon's trying to say is, come on guys, think of some original questions.
No, I think people are terrified of the cell phone idea, I think. But the quality control issues with that particular approach are at least 5 years away. We have no idea what happens on an individualized basis, as I explained before. So there's a lot of QC issues that have to be ironed out with regards to that technology, at least.
Okay, yeah, I mean, other views on how quickly some of this new approach is going to have an impact?
Okay, so there are treatment types of Parkinson's with genetic origins like LRRK2 or GBA. So those might be the first to come along, where we might hope, if they work at all, that they'll work clearly in that genetic subset. There may as well be tests to indicate if someone has low or high enzyme activity, that they might be especially good or poor candidates for one of those treatments. So those treatments are in clinical testing now. And I can imagine, at least in a few cases within 3 to 5 years, we may be able to say, here's a new treatment that we think will work actually only in 15% of people with Parkinson's, and this is how we detect them, and you can get that test. But that would be partial with a few treatments. I don't think we're close to the point where every single individual can be profiled and given the customized therapy.
I completely agree. I think we will first be, let's say, chipping away at the edges, right? So, and that is already happening. There are already drug trials which are currently, as we speak, carried out in this country, which only include patients with Parkinson's disease who have a particular genetic defect, so to speak. And that really does make an awful lot of sense for the drugs which are currently being tested. So I think I'm very certain that we'll be seeing more and more of this. But the next big step will, of course, be, how can we translate any results from these very focused trials on comparatively few patients to the much more common sporadic form of Parkinson's disease. That's going to be the big challenge.
Yeah, I mean, you've talked quite a bit, Oliver, about genetic and non-genetic symptoms and the progression.
So to cut a long story short, there aren't really. There are gene defects which typically lead to early onset Parkinson's disease, such as mutations in the Parkin gene, and there are other genetic mutations which typically lead to later onset Parkinson's disease. But if you compare patients with Parkin mutations who develop the disease at an early age to other patients who develop Parkinson's disease at the same early age, then it's really not possible to tell the difference between those who've developed the disease due to Parkin mutations and those who haven't. Certainly not when you just look at the individual patient.
Thank you. And it's great that we're getting some questions from people who are watching online as well. And there's a question from Teesside, so that's exciting. Is there any connection with this research and the theory that Parkinson's starts in the gut? Simon, whether you want to...
So this is the third most common question I get. How is it that something that's going on in the brain could be coming out of your breath? An obvious source is either the lungs, or the gut. And remarkably, Professor Hayek's team see a very consistent pattern when we take multiple samples. They see a very consistent pattern within individuals. So it doesn't matter what you ate for dinner last night, we still see the same pattern. So that sort of goes against this idea that it could be food or drink related. It doesn't take anything away from the idea that it could be some bacteria in your gut that is producing these particular chemicals. This is obviously the next step in the research, is to determine exactly where in the body it's coming from. And there are certain ways we could do that. And we're certainly investigating the best approach.
Brilliant, thank you. How does a medicine for one condition actually make a difference in another? Arthur, do you want to have a stab at that?
Yeah, well, I guess there are a number of ways. I mean, I'm sure Oliver will have thoughts on this as well. In some cases, we find that a drug that was developed with a particular purpose in mind and achieves that purpose by acting on a particular protein in the brain and the cell actually is discovered later to act on another protein as well. So there's a drug that I'm very familiar with called sulfonamide that was originally developed for epilepsy, actually, as a sodium channel blocker. Years later, it was discovered that it's actually about 50 times more potent, 50 times more active on MAOB, the enzyme that rasagiline acts on that Oliver referred to. So this drug was already in hand, the drug was already tested, the drug was already known to be safe. And at that point, it was discovered to work on a protein which is known to be involved in Parkinson's and known to bring some relief to Parkinson's. So at that point, the company involved switched and said, let's go for Parkinson's because, in fact, it's not working so well in epilepsy. So there's a case where there was a good scientific rationale for it. In other cases, it's more empirical, where there's simply, as you alluded to, there could be epidemiological data. Large populations of people with one disease that have been taking a certain medication also seem to have more or less problems with Parkinson's, and that leads to the idea that you could repurpose a medication. So there are a number of routes to it. It's more common than I would imagine. In Parkinson's, there are a few treatments, including selegiline and including amantadine, which are repurposed, were originally developed for one medication, and now we find they're useful in Parkinson's. Yeah, Oliver, anything to add?
Yeah, it's a fascinating area, and the main reason why academic clinicians like myself or Kamal Carroll down in Plymouth or Caroline Williams-Gray here in Cambridge are so keen to now repurpose drugs which have been in clinical use for decades for, in Parkinson's disease trials, is because it's so much quicker and so much cheaper and so much safer compared to us trying to take a completely new compound first into what's called phase 1 trials, which means that, this is a trial which only includes healthy people, and the first thing we then need to find out with a completely new compound is whether actually healthy controls can tolerate this drug, and that's a hugely expensive step in drug development. And we don't need to do that with UDCA or statins or azathioprine or whatever repurposed drug we want to test in Parkinson's disease. We know these drugs are safe, or at least we know a lot about the side effect profile. And that in its own right makes it more attractive. However, I also, have to say that was one of the main reasons why I struggled so much to get money to take UDCA into clinical trial, because let's face it, if nobody can make money from a drug, then people are just less interested in supporting clinical trials. And that's a fact, right? And I'm afraid this doesn't just apply to drug companies, it also applies to funding organizations.
Okay, thank you. So a question here, someone's saying how their husband was diagnosed less than 2 years ago. Should one ever consider it's too late? The moment has been passed to kind of identify what type of Parkinson someone has had. I guess it was you talking, Oliver, about that screening at the point of diagnosis. Can that happen for someone who's had Parkinson's for longer?
So I want to make quite clear again that I'm at times a little over-enthusiastic, okay? So I think our data are exciting. I think our data and the data from many other groups hold great promise, but at the moment we don't know yet whether any of the concepts we discussed today, any of the concepts that are published in the literature will really pan out, right? So That's why we just need to be a bit skeptical. But my prediction is that it will not matter all that much whether we do all this assessment, breath analysis, the genome analysis, the clever brain scans, and the analysis of the skin cells, and so much else. Whether we do that at baseline, so to speak, or 2 years into the illness, I don't think it's going to matter a great deal. I think once a patient's had Parkinson's disease for 10 years or more, it might become very difficult, very hard for these drugs to still rescue the remaining nerve cells. But in the first few years or so, I don't think it's going to matter all that much.
Thank you. So just a couple more. One person's saying, if I see my consultant every 6 months, why is he not gathering the information needed from me already? So this sense of people with Parkinson's have got so much information to share with the research, the clinical community. How do we encourage that and really make sure that we're accessing all of the data that's readily to hand at the moment? Does anyone want to comment on that challenge?
So I'm wearing lots of different hats, okay? So I'll first answer this question with my NHS hat on, okay? So I do 2 clinics every week, one in Sheffield, one in Doncaster. I have 15 minutes per follow-up patient, and in that time I'm also expected to dictate the letter, right? It's not a lot of time, and that time is easily taken up by talking just about the very problems the patients developed over last year, the things we badly need to tweak, okay? So I just don't think it's ever going to happen within a regular, routine NHS appointment. Forget about that. Now put on my researcher's hat and say, and really thank you to Parkinson's UK for helping us with this. We've now got a fantastic PPI group in Sheffield, and that's the way to do it. You need to identify patients and relatives, spouses and so on, who've got a particular interest in Parkinson's disease. So a few weeks ago, we had a bunch of about 15 of them at Citron, and they just gave us advice. We explained our drug trial to them, and we got advice from them. And I'll tell you one thing, it was fantastically useful. And they raised so many important points. And I think that's how we need to do this. We need to ever increase the interaction between patients and the relatives and the scientists, but we need to do it using different structures. We need to move it away from the routine clinical appointments.
Brilliant, thank you. Any other additions to that? Okay, let me ask a final question. I mean, it's fantastic that this lecture has become an annual event. This is the 9th, next year the 10th, and we'll keep on doing this. So and you've been brilliant tonight, so we'll invite you back for a reunion on the 15th anniversary, so in 5-6 years' time. Just it'd be great to hear all of you just very briefly say, what do you hope that we are talking about in 6 years' time? What's the kind of message? What's the sense of where Parkinson's at? I guess in what you're hoping. Arthur, do you want to go first, or we'll perhaps move down the panel this way?
Well, so I think I was just at a meeting today of the Multiple Sclerosis Society talking about new treatments in the multiple sclerosis area. So they have to recognize that they're fortunate in that there are now 10 disease course modifying treatments that are approved for use in multiple sclerosis. So when a patient is newly diagnosed, they actually have a range of choices. They have some gentler, easier ones people start with. They have some stronger and perhaps riskier, stronger medications for people that need extra relief. And it's a matter of making the choice, the right medication to start everyone on, and seeing if you have. So I think if Parkinson's in 6 years which is the right medication for which person, disease course modifying treatments, that would be fantastic. I think it's not that far away, whether it's 6 years or 6.5, perhaps slightly long. I'm not sure.
Okay, thank you. Ruth, what's your vision?
6 years would be great if I and all my friends who have Parkinson's could say, we feel better, you found stuff that makes us better, you've stopped it, we've actually recovered some of our brain cells. That's fantastic. Whether it's feasible, I think, yeah, why not? Let's say it's feasible, because you have to be positive, I think.
Brilliant, thank you. Simon?
So in 6 years' time, I actually suspect, I'm quite optimistic, and I suspect we're going to be sitting here talking about a dilemma, which is, and it comes to the very heart of the idea that we need better diagnostics and better assessment tools, because we're going to have semi-curative treatments in the very near future, I believe, that everybody in the community will want to jump onto, and this is going to cause a big dilemma for future development, because everyone will be on that drug, and that drug will be an exclusionary for any future trials, because the beneficial effects of that drug will be difficult to, maybe there are folks who have other opinions on this, but so I think in 6 years' time we're going to be having sessions like this where we're going to be talking about very specific biomarkers that allow us to measure the beneficial effects of a new drug while everyone is taking this other drug.
So
I think that's a good
problems.
It's a good dilemma to have, yeah.
Okay, yeah, and Oliver?
So, in 6 years, we will have cracked one essential problem, which is holding us back more than you can possibly imagine. There's been fantastic progress at the preclinical end, right? Research, the progress has been phenomenal, believe me, at the bench, right? But we're still rubbish at measuring disease progression in Parkinson's disease, right? The drug trial protocols we're using at the moment are just as poor as they were 10, 20, 30 years ago. And that's a big, big problem. But things are moving, and they're moving fast. So in 5, 6 years, we will have much more robust clinical trials protocols which will allow us to test promising drugs much more quickly in smaller groups of patients. And that's going to be the key. And I often compare the situation I imagine to what happened with HIV. So when I was a medical student, the very first drug treatment for HIV was introduced, and it had next to no effect, right? And now patients who are HIV positive are on a drug cocktail which has really transformed their lives, and they now have a largely normal life expectancy, I believe. So that's what we're going to see. The first few drugs which will come through will not be the magic wand, but eventually people will be on a drug cocktail which will change their lives.
Fantastic. And I think that's a really optimistic note to end tonight's series of lectures. When Ruth kicked the evening off, it was only, I think, her second sentence where she used the word hope. And hope is such a driving force, isn't it, amongst the Parkinson's community. It was in her third sentence she introduced the sense of a community. And what I get out of tonight's series of lectures, that it is that Parkinson's community with all of its parts that is going to be instrumental collectively in delivering the vision that we've just heard. It's our fantastic research community working at a real kind of cracking pace of innovation. It's the breath machine that is now small enough to be on a mobile phone. It's that ability to create to take skin cells and do fantastic drug screening on all of that. And all of this technology is going to be accelerating, and we want to make sure that it's deployed in the field of Parkinson's rather than others. So continuing to support our fantastic researchers. There was a sense there, I think, about working internationally and forging those partnerships, working with Israeli scientists, the work that Parkinson's UK is doing with international partners to try and change the way that clinical trials are being planned, which is so key. There was a sense here in which the voice of people living with the condition is going to be really important to shape the way these treatments develop, some of the ethical questions that play. Whose data is this that we now have? How do we collectively use this data and make sure that it's used in a really responsible way to drive forward those kind of treatments? People living with the condition shaping clinical trials, informing the researchers of what the priorities need to be. So much of the research, all of the research that we heard tonight, funded by Parkinson's UK, and let's not take that for granted. That's money that we need to get out and raise every year. It was fantastic talking to people this evening, local groups saying this week on World Parkinson's Day, we went out there and we raised £950, and that's going into research. That's the reality of how this happens. It's people going out of their way, running marathons, digging deep into their pockets to fund this, because that's the currency of hope. It's people who are volunteering in research studies, breathing into machines, giving their skin samples so that research can be done, being podded and poked and asked to fill in mood questionnaires and all of that. So there is a sense here that hopeful vision that we heard for 6 years no one else is going to deliver that other than people in this room, people online, and that global community of people affected by Parkinson's. And I guess that's our call to action. So I really want to thank the speakers for their fantastic presentations and leave us all with that sense of, come on, what can we deliver in the next 5 and 6 years? So I want to say thank you and goodbye to our online audience. I hope the technology has worked and it's been a really positive experience.